Greetings and welcome to the United States Transhumanist Party Virtual Enlightenment Salon. My name is Gennady Stoliarov II, and I am the chairman of the U.S. Transhumanist Party. Here we hold conversations with some of the world's leading thinkers in longevity, science, technology, philosophy, and politics. Like the philosophers of the Age of Enlightenment, we aim to connect every field of human endeavor and arrive at new insights to achieve longer lives, greater rationality, and the progress of our civilization. Greetings, ladies and gentlemen, and welcome to our U.S. Transhumanist Party Virtual Enlightenment Salon of June 18th, 2023. I'm very pleased to welcome you today to a wide-ranging interdisciplinary conversation with a fascinating guest, Logan Thrasher Collins. But first, let me introduce our distinguished panel for today. We have our current Vice Chairman and Director of Visual Art, Art Ramon Garcia. We have our Director of Longevity Outreach, Ben Ballweg. We have our Legislative Director, Jason Geringer. We have our Technology Advisor and Foreign Ambassador in Spain, Dr. Jose Cordero. And our guest of honor today is Logan Thrasher Collins, who aims to make science fiction into reality. He is a futurist, synthetic biologist, author, and innovator, as well as a 2023 Foresight Fellow. He is also a prolific science fiction author and poet. When he was 16, he invented a new antimicrobial protein, OPAL, overexpressed protein aggregator lipophilic. He next developed a bacterial conjugation delivery system for the gene encoding OPAL. His synthetic biology research has been published as a first author journal article in ACS Biochemistry entitled Design of a De Novo Aggregating Antimicrobial Peptide and a Bacterial Conjugation-Based Delivery System. He is currently a PhD candidate in biomedical engineering at Washington University, St. Louis. Welcome, Logan, and I understand you have a presentation for us, so please begin.
Great. Thank you very much, Gennady. That was a wonderful introduction, and I'm really happy to be here today to present sort of a story of my path so far, as well as some more about my current research and some future ideas that I have for connectomics, or mapping the brain. All right, I'm going to start sharing now. So just let me know if this looks good. I think this should be visible to everyone. So today we're going to be doing an enlightenment salon with me, Logan Thrasher Collins. And I'll get started by just telling my story. And this will provide everyone with sort of some groundwork in who I am, what I've done, and the types of things that I'm interested in. All right. So, as was described in my introduction, I'll talk a little bit about my past research on developing new ways of combating antimicrobial resistance. But to start, I'll just say I've been a lifelong lover of science and always a big dreamer. Ever since I was super young, I've always known that I really want to do something to make a substantial positive impact in the world. And following this type of passion, I ended up starting scientific research in an academic laboratory at the age of 16. So I actually came up with an idea for this research sometime before that. So it was actually eighth grade, which I suppose would have been something closer to the age of 13. But I spent a little bit of time developing it, and then eventually joined a lab a few years later. So this was an amazing adventure for me because from the beginning of that research, I ended up inventing what Gennady mentioned in my introduction, the OPAL peptide, as well as a bacterial conjugation-based delivery system for the gene encoding that peptide. This took me on a range of amazing experiences, from being able to speak at TEDx Mile High when I was 17, to winning Best of Category at the International Science and Engineering Fair, as well as the Dudley R. Hirschbach Award, and being able to take a trip to be present at the Nobel Prize ceremonies in Stockholm, Sweden. So that was really my high school experience. Really, a lot of amazing science adventures, being able to really see sort of the pinnacle of the scientific world, as well as take part in creating something entirely new and doing research for the first time. To talk a little bit more about what I did, you'll see in the image here that there's a couple of bacteria. Basically, what I did was I invented a way of creating donor bacteria that could transfer a piece of DNA to recipient bacteria, and then for the recipient bacteria to be able to turn on a gene that I designed that then produces the antimicrobial opal peptide, which aggregates, disrupts bacterial homeostasis, and kills off the recipient bacteria. The nice thing about this is that the turning on of the gene can be targeted to specific types of bacteria. So, in this way, it can serve as a platform technology for precisely having donor bacteria, which don't turn on the gene, delivering this gene to target recipients, which do turn on the gene, and then sort of precisely just pruning the population of bacteria to remove the bacteria we don't want. In the context of a potential future therapeutic, this is really a positive thing because it means that you could hypothetically deliver donor or put donor bacteria at the site of an infection where they would deliver this gene to both your indigenous microbiota as well as to whatever bacteria are causing the infection. And then it would only kill off the bacteria causing the infection. And I was able to verify all of this experimentally in vitro. In vivo, it has yet to come, but I do have aspirations of continuing this in the future and developing perhaps a spin off company from it once I have the resources to do so. Uh, just to provide a little more explanation of this microscopy image, you can actually see the aggregates caused by the opal peptide here. So it's really cool. Basically, I wanted to be able to verify that it actually was causing aggregates, and so I ended up having this really using it was, I believe it was structured illumination microscopy to, or maybe it was just confocal. Anyway, it was a while back, but the microscopy was I used a stain with a dye called Nile Red that stains hydrophobic aggregates as well as other hydrophobic molecules. And because I designed the opal peptide to be highly hydrophobic as part of its aggregation mechanism, I ended up being able to visualize it directly in this fashion. So really cool. This was my intro to science, and it really just set me off on this path of loving science, loving inventing things, doing research, etc. For part two, I'll really be covering my college experience, so undergraduate years. And this is when I really got interested in some of the stuff that I'll talk about a little later, which is connectomics and whole brain emulation. So, as since this is the United States Transhumanist Party Enlightenment Salon, I assume many of you may be familiar with whole brain emulation, but just for anyone who isn't, whole brain emulation is essentially the idea of recreating a brain in a computer simulation with a sufficient level of detail that it actually has some form of its own consciousness. And this is oftentimes coupled to the concept of mind uploading. People will perhaps say, well, you could gradually replace parts of the brain, maybe even a whole hemisphere at a time, and you would be able to continually transfer someone's consciousness into a computer, thereby paving the way for very futuristic goals of things uh, uh, von Neumann machines with simulated or emulated human minds traveling to other planets, people living for a very long time in virtual worlds, et cetera, et cetera. Very very inspirational stuff. Very perhaps in some ways further future stuff, but things that I find to be highly compelling uh, as a form of envisioning what life could be like in the future. And I'm perhaps someone who is a little bit more cautiously optimistic about this. So I'm not envisioning things like mind uploading happening anytime in the near future, but I think it's possible, and it's definitely something that we should strive for. So how did I get into this? I actually, perhaps ironically, saw a science fiction film, which was an episode of, surprisingly enough, Black Mirror. Um, it was called San Junipero, and for those of you who aren't familiar with it, it was essentially the only optimistic episode of Black Mirror. So normally, I'm not a huge fan of Black Mirror because I feel that it perpetuates certain negative stereotypes about technology, and it stirs up people's fears in ways that maybe aren't entirely fair and not entirely balanced. Uh, there's arguments to be made either way, but that's just sort of my personal take on it. That said, I found this particular episode, which was more optimistic, to be highly compelling. It told the story of a couple of lovers who were actually very old in real life, and they ended up pursuing an afterlife together in a supercomputer, basically, which is pictured up top. And this is sort of the vision of these lights representing the different souls of people, sort of glittering in this very beautiful array. Um, so I found this to be a very very stirring vision, very compelling vision. And so I started just reading up and delving into this field, which up until that point I hadn't really had a huge amount of knowledge on. Um, but I really dove into the literature, and I actually ended up, as I was wrapping up my other project with the antimicrobials and publishing it, I also began doing research in connectomics, mapping the brain. So to start, what I did was I developed a something called an immunogold stain, which is essentially a way of staining particular structures in the brain, and was able to show that X-ray microtomography can reconstruct in 3D tissues that have been stained by this immunogold. I specifically used gold nanorods with antibodies linked to them. I was able to link the antibodies on and then target the gold nanorods to a dopamine receptor and stain things like a medium spiny neuron as well as some other neurons in the tissue. You can see the 3D reconstruction here. You can also, I also have images of this that have been sort of rotated so you can see the side of the tissue slice. Anyway, very cool stuff, but sort of very preliminary, at least from my current view. And there actually has been some work that was published on a similar study that sort of superseded what I did later on with that particular thing, by a different group. However, I ended up doing this for my one of my two senior theses of during undergrad, and it's available. The study is available in the database of the university if anyone's curious. But that I feel like was really just my intro to this world, because I have a lot of other ideas on it. Um, I ended up conceptualizing some early ideas for combining X-ray microscopy with expansion X-ray or with expansion microscopy. Expansion microscopy is essentially enlarging tissue in order to, in an isotropic way, a way that preserves spatial relationships, such that it allows for making everything bigger so you can see things more clearly. And I'll talk a lot more in depth about this later, but I began to conceptualize some of these ideas, do a few early experiments, and I ran into essentially a problem of signal dilution, which made it so that I wasn't able to see a whole lot. However, there has been a recent paper published or a recent preprint published that actually I believe will solve this problem, just so long as I'm able to actually get the resources necessary to do the experiments, which I don't currently have access to, but I do have a group that I'm currently collaborating with that may be able to perform some of those experiments in the future. Anyway, I'm getting a bit into the weeds here on this because I'll be talking more about it later, but um, basically the take home message is I began to lay the groundwork for some of the things that I'll talk about later and some of the things that I hope to pursue in the future. I also published a perspective paper that was actually a sole author paper on insect brain emulation and the path to essentially doing whole brain emulation of insects as a first step towards later whole brain emulations of mammals and then eventually humans. And in the insect brain emulation paper, I actually made a somewhat bold prediction of 20 years from now, there being the possibility of insect brain, really realistic insect brain emulation that could recapitulate the behaviors of insects in a virtual environment in a realistic fashion. And I think that could be either a great underestimate or a great overestimate, depending on how much resources is put into this particular scientific question. So I'm not saying that that's a hard line at all, but it was just sort of a rough outline that I suggested. So I'm definitely a little bit more of a long timeline person when it comes to this stuff at this point, but I do think that it's possible. And again, I do think it's a vision very much worth pursuing. All right, so for part three, I will talk about my experiences sort of shortly after I graduated from college. So I was working on all this stuff with expansion microscopy and X-ray microscopy, and then the pandemic happened, and the labs all shut down. Everyone was home, everyone was scared, and I wanted to do something to help. So I joined a startup company called Conduit Computing, first as lead scientist and then eventually as CTO. I was sort of the acting CTO all the way through, though. Um, uh, Even as lead scientist, I was sort of doing the job of the CTO. Anyway, I joined this small startup called Conduit Computing, which was founded by Ryan Robinson, CEO. And Ryan is a really great guy. He designed his own major at MIT. He calls himself the world's first quantum engineer, and he really has his foot in a lot of different places, like myself. And so when I connected with him, we felt a great synergy, and we wanted to work together on this problem. Conduit had had a history of working with high performance computing or supercomputing for financial calculations, collecting supercomputer resources and then lending them out to different people who wanted to do work in finance. But Ryan really has always had, as have I, a vision of doing something a little more humanitarian. And so what we wanted to do was we wanted to do sort of this nonprofit approach uh, uh, to fighting the pandemic. And so what happened was we found this opportunity called the COVID-19 High Performance Computing Consortium. And that's essentially a coalition formed by the US government, the White House, Department of Science and Technology, as well as some other organizations, which brought together both academic and industrial supercomputers on a scale that was even larger than what Conduit had done previously to lend them out to people who were working on essentially computational approaches to fighting coronavirus. So what we did was I wrote this proposal with a little help from Ricky Williams, another member of Conduit, and we submitted it to the consortium. It was accepted, and we gained access to the Frontera supercomputer, which at the time was the fifth fastest supercomputer in the world. Since then, it's dropped down on the list since people have built even faster supercomputers. But at the time, it was in the top 10, and it's still a very powerful computer, obviously. So what we did was we were studying a process, and this was my sort of investigation into a somewhat niche area on purpose because I wanted to explore something that not a lot of people were looking at. We investigated a process of coronavirus's life cycle called budding. And budding is essentially how it wraps membrane around its genome in order to form complete coronavirus particles inside of the cell. So, in order to package its genome, it has to steal a part of an organelle's membrane called the ER Golgi intermediate compartment. And to steal this part of the membrane, it has to induce curvature in the membrane. And to induce curvature in the membrane, it's known that certain proteins called M and E proteins are involved, but it's not fully understood how they're involved. And so basically, what we did was we used molecular dynamics simulations, six simulations consisting of over 500 or around 500,000 atoms each. One of them is pictured here. A snapshot of one of them is pictured here in the publications screenshot. And we ran these simulations to see how the proteins would interact, see how they would interact with the lipid membrane. And we established some interesting biophysical findings that may have implications for potential outside the box ways of treating coronavirus infections in the future. We published our findings in the ACF's Journal of Physical Chemistry Letters, and you can see that publication here. And so now we've contributed to the scientific conversation. People have actually cited our paper, and they have built upon the results of our models to create even more elaborate computational models that have derived further insights. And so we've really been able to establish something that has actually moved the field forward and may someday lead to to treatments not just for the current types of coronaviruses out there, but for other types of coronaviruses as well, not just the current variants of SARS-CoV-2, that is. Because these particular proteins are actually highly conserved among different coronaviruses, even different species of coronaviruses. So that was Conduit, and that actually took place partly during my graduate school experience, first couple of years. I've also worked at Conduit on a project called Nanosplash, which is still in its early stages, but it's essentially a diagnostic for testing people for infectious diseases, not just coronavirus, but other diseases as well, such as tuberculosis. And that project is still in the early stages, but I'm helping them along, and I was the originator of that concept, and we actually patented the conceptual design of Nanosplash. And so that's another thing that's going on with Conduit. Moving on to my graduate school experience. So, as was mentioned in my introduction from Genedy, I'm now a biomedical engineering PhD candidate at Washington University in St. Louis. And during my time so far, I've been doing a lot with writing grants, with performing lab work, of course, at the bench. And I've also published a couple of review papers that you can see that are really forward-facing review papers in ACS Nano and ACS Synthetic Biology. In particular, the one in ACS Synthetic Biology was what I would call a visionary, perhaps somewhat without humility there, but what I would hope would be considered a visionary review paper, in the sense that it's really more of a perspective on what the field of AAV, adeno-associated virus manufacturing, can do to make adeno-associated viruses, AAVs, much more accessible to a broader range of people. Because right now AAVs are really hard to make, and these manufacturing costs contribute to their extremely high costs of the therapeutics that are derived from them. And this means that gene therapy in general, since AAVs are among the most popular gene therapy delivery vehicles, gene therapy in general is being kind of held back from really being able to be distributed to everyone. And as transhumanists, we really want gene therapy to be something that's commonplace enough that there can be a lot done with it. And so what I'd like everyone to be able to access AAV therapies and gene therapies in general, but this paper focused specifically on AAV. And so I propose synthetic biology methodologies for AAV manufacturing. And if you're curious, you can read more about it in that paper. But what am I actually working on beyond just speculative stuff here? My PhD thesis project, I actually have 3 different projects I'm working on currently, but 2 of them are proprietary, and they're in collaboration with other groups, and I can't really talk about them, but I'll just say they're really cool, especially one of them. And once I get IP on those with David Curiel, who's my PhD advisor, we will be able to speak about them more openly. But for now, I'll talk about the one that we do have IP on, we do have a provisional patent on, which is the AdAAV project. So that's what you'll see up here in this top set of images. And I'll be talking more about this shortly, but it's essentially a way of delivering multiple genes to the same cell, broadening the scope of gene therapies in such a way that very complex genetic engineering could potentially be done in humans at some point in the future. This is actually the subject of one of the 2 segments of my presentation that are coming shortly. It's the first one. And then I'll also mention that I've published several science fiction short stories and poems. Writing, really story itself, has always been a big part of my life, and keeping in touch with my artistic side, keeping in touch with um, bringing together the sciences and the arts has always been something that I've been very passionate about. And so I am just always writing science fiction and poetry and so on and so forth because it really feeds my soul and it helps me to envision the future in positive ways that can, as well as ways that interrogate the potential issues that may be encountered along the route. And with that, I guess I'll transition into the next part of my presentation. Uh, this introduction took longer than I expected, so my apologies for that. But hopefully, you guys will bear with me as I talk a little bit more about my gene therapy research. So, my gene therapy research, specifically the AdAAV project, centers on the idea that adenovirus and AAV, these are the two viruses pictured here, they're both leading technologies for gene delivery in a therapeutic context. However, they come with benefits and drawbacks, and there are ways to combine, or sorry, I'm developing a way to combine the two in such a fashion that the benefits are enhanced, the drawbacks are mitigated, and the potential for delivering multiple or even many genes to the same cell becomes realized by literally physically sticking the AAVs onto the surface of the adenovirus using a technology called SpyTag SpyCatcher. And so, yeah, this could lead to treatments for numerous diseases such as Alzheimer's, hepatic cirrhosis, even complex ailments like obesity and cardiac diseases, so on and so forth, arteriosclerosis. And I do have a vision for perhaps someday this type of technology contributing to helping to introduce radiation resistance pathways into astronauts so that they can survive and thrive in outer space and on other planets more in a better way. And so I'm really excited about the potential of this technology. Currently, it's definitely an ongoing project, but it's something that I've made some significant progress on, and so I wanted to share today. The enabling technology here to stick these viruses together is called SpyTag SpyCatcher, which I mentioned before. Essentially, there are a couple of small proteins or peptides that react rapidly and specifically with each other to form a covalent isopeptide bond, which is essentially a bridge off of the main polypeptide chains that leads to that is a covalent bond, so it's very strong. And it allows for proteins to be engineered to be fused to the spy tag and the spy catcher so that two things can come together in a way that basically permanently links them unless you really provide a lot of energy to rip them off. I won't go into all the details of this particular slide, but I just want to say that one of the main challenges that I had to deal with was essentially finding a way to build these things without creating massive aggregation or having too many byproducts. Essentially, leftover AAVs it was the biggest issue. AAVs just floating around that had not been connected. And so I developed a novel assembly and purification method in the lab to produce AdAAVs. And the steps of this method are shown here. I've made sure that this works, and it does, and it's cool. Essentially, it goes through this process of functionalizing, mixing, and then purifying the final product. And I'll show you, there's sort of three different experiments because first I just wanted to make sure that I could make these things without purification. So when you start, the reason there's no contaminants here is because I diluted these a lot. But when you start with AdAAVs that are without the dilution, you would see a bunch of AAVs floating around in here, as well as a few aggregates of multiple adenoviruses. But essentially, what you can see, and this is transmission electron microscopy images, so you're actually looking at what these actually look like, which is really cool. Essentially, what you can see is that when the spy tag is available on both viruses, I'm using a bi spy catcher to link them. When the spy tag is available on both viruses, you do get these very nice add AAVs with multiple AAVs per adenovirus, maybe around 5 to 10, depending on how lucky you get for the particular specimen. However, when the adenovirus does not have spy tag, occasionally you'll see sort of like maybe an electrostatic association between the adenovirus and an AAV, but by and large, there's not the same type of linkage. So this validates that the first step can be accomplished. And then I actually did a small batch purification for my second my second central experiment for this series of steps. But I'll skip that since I didn't include that in this presentation. Then eventually I did a large batch purification. The small batch purification, I'll just note, was basically to make sure that this biotinylation as an affinity reagent would work correctly using the streptactin affinity column, but that's kind of a technical detail that I think would take too long to explain. So I'll just say that eventually I was able to purify this mixture, which you can see the pre-purification assembly mix is quite messy, and obtain add AAVs in a relatively pure form where you can see that they still look like add AAVs and are correct. I've also titered these via qPCR, and I've begun preliminary cell culture experiments for actually infecting cells with AAVs and seeing how well they are able to transduce them or how well they're able to actually express the genes that they're delivering. And that's all still in progress, but I'm definitely making some progress, although I have been working on some of the other projects right now that are more that are proprietary, and so I've taken a pause on some of these things, but I'm going to get back to them in just a couple weeks and hopefully make some further breakthroughs that would be really cool to show in a presentation like this if I had them already, but unfortunately I'm still in the ongoing stage. Nonetheless, it's all very exciting, and I am hoping that this will go far. All right. So finally, this is the last section of my presentation, and then we can get to the discussion section, or the like question and answers and discussion. I want to talk about a little bit more about my future ideas for connectomics. So connectomics, mapping the brain, is a field that is sort of plagued by a speed issue. So yes, it's possible to reconstruct parts of the brain in extremely high detail, potentially high enough detail to, at some point, translate this information into whole brain emulations. Though I should mention that that comes with the caveat of that's perhaps an even bigger engineering challenge. And I'm actually working with Randall Cohn and Ken Hayworth on a paper, which is a forward-facing perspective paper that will propose some potential ways around that. But essentially, my focus as an engineering person, as somebody who wants to solve the problems in the order that they are coming at us, I guess obviously there's something to be said for parallelism, but that's why there's multiple people working on different things. My particular area of interest is more in connectomics, so actually mapping the brain. And I'll just review the field so far. So there's sort of two big approaches that are being used currently. Electron microscopy, which is very, very slow. So for example, there was this big effort by Yin et al. To image a cubic millimeter of brain tissue. It took 6 months of data acquisition using 6 fully automated serial section transmission electron microscopes. Each instrument cost about $125,000 for a total of $875,000. And this was the size of their voxels in the images, 4 by 4 by 40 nanometers. Very high resolution, but not isotropic. So not isotropic just means that all the dimensions are the same value. So like if it was 4 by 4 by 4 instead of 4 by 4 by 40, it would be isotropic. So I did the calculations, and essentially it would take 3.6 million years divided by the number of microscopes, or 600,000 years using the setup that Yin et al. Had to map a human brain with these numbers, because the human brain consists of approximately 1.2 million cubic millimeters. So yeah, there are some people working to develop faster electron microscopes, but the main goal at this point for the field is the mouse brain. and the human brain is 2,400 times more volume than the mouse brain. So even if people were able to create a massive foundry where they parallelize hundreds or thousands of electron microscopes to map the mouse brain, uh, I'm skeptical that they would be able to scale up to the human brain anytime soon. There's also a method called expansion light sheet microscopy, which is essentially something that's relatively early in the development process compared to electron microscopy, but has some promise. And I calculated based on this fairly recent publication in which Lilvis et al. They imaged a small part of the fly brain called the central complex, which essentially had a volume of only about 0.001 millimeters cubed. Uh, and what I found was they just used one lattice light sheet fluorescence microscope. They made it 8 times bigger using expansion microscopy, and they imaged in 3 colors. And it took them 5 days to image that with 15 by 15 by 50 nanometer voxels. Um, that was an approximate estimate based on their resolution of 30 by 30 by 100. There's a relationship between resolution and voxel size that you can approximate by doubling it, but there's also other things that go into it. You can look into it. It's called Nyquist sampling. There's more to it. Anyway, the microscopes, the lattice light sheet microscopes currently on the market, as far as I know, cost around $700,000. It might be possible to get that to be less expensive in the future and even have sufficient. Sorry, been talking for a while. Even have significantly better microscopes. However, hang on just a moment. the the light sheet microscopes actually took even longer based on my calculations, would take even longer based on my calculations to map a human brain, 16.8 million years divided by the number of microscopes, which honestly I'm kind of surprised by because generally speaking, light sheet microscopy is considered much faster than electron microscopy. So maybe it was just an idiosyncrasy with this particular paper, even though the paper was titled something like Rapid Reconstruction of Neural Circuits Using Lattice Light Sheet Microscopy, emphasis on the rapid. But suffice to say that it does still seem like there might be a speed issue here, and even though there may be substantial improvements coming soon by an organization called E11 Bio, E11 Bio is seeking to use a similar approach to map the brain, probably first the mouse brain, as using light sheet microscopy and expansion microscopy, and they've made some claims that they'll be able to do it in a reasonable time frame, but it still will require parallelization with thousands of microscopes and hundreds of millions of dollars, which I don't think is really the best route, personally. There are some benefits in that you can track multiple colors more easily and so on and so forth, but anyway, this was all set up because I want to present another route, synchrotron X-ray microscopy. It has some great advantages. It's non-destructive, so that means you don't have to slice up the tissues very much. You can section them into chunks. To some degree, you'll need to section them into chunks, but the chunks can be quite large, so less alignment issues between slices. It's isotropic, so that means that the voxels are the same dimensions in X, Y, and Z, and therefore the computational reconstruction of the tissue is easier. And it's also very, very fast compared to all the other methods. I'll discuss this in subsequent slides. It is possible to image in 2 to 3 colors. There might be some possibilities for imaging in more, depending on whether someone can come up with really cool physics ways of doing that. The disadvantages is that it requires a synchrotron facility beamline, and a synchrotron is a giant particle accelerator. And the beamline is basically a part of that particle accelerator that sort of sticks off at a tangent angle to the main storage ring and has some equipment in it to basically insert X-rays that have been generated by the storage ring from the acceleration of electrons around the ring, and the deacceleration braking. There's a whole big relativistic thing that goes on where they're able to basically emit this extremely powerful X-ray beam from this this ring of electrons being circulated around. And they use an insertion device. There's several different types: bending magnets, undulators, wigglers, and there's various differences between them. But the point is that it emits a very powerful X-ray beam. Um, and then essentially you do some optics and it passes through the sample. It goes through a scintillator to convert it to visible light. And then there's a mirror and that projects it through a lens system onto a detector. So that's how it works. And to do it in a tomographic fashion, you use a rotating stage so that you can rotate it around 360 degrees during imaging, taking different projection images, flat projection images, and then using some fancy maths to reconstruct a 3D reconstruction of the brain tissue from there. So that's the synchrotron beam facility beamline and how it works. There are laboratory scale X-ray microscopes, but they're not bright enough to image the brain efficiently. The resolution in general is not enough for nanoscale connectomics, assuming you're wanting to image with any reasonable level of speed. So synchrotron X-ray microscopes can't image fast enough if you're going to go down to like super high resolutions, but they can if you're at lower resolutions. Like 300 nanometer voxel size seems to be sort of the best people can do if you're imaging millimeter scale stuff in seconds or minutes. However, expansion microscopy. So, as I've alluded to several times throughout this presentation, expansion microscopy is a way that you can physically enlarge tissue in a way that preserves spatial relationships. And the way it works is that you infuse tissue with a swellable hydrogel. You cross-link everything to it. You do some digestion to sort of loosen things up. You label things, and then you put water on it, and it blows up basically, and it gets bigger while staying the same shape. Various methods can achieve various degrees of expansion. 4x, 10x, 20x, 25x seems to be sort of the biggest that they've been able to do so far. There's something called iterative expansion microscopy, which achieves the 20 and 25x. Basically, you do it twice. You relink to a second hydrogel in the second round. Some of the newer methods can label membranes, which is important for connectomics, for morphology of the neurons. And a lot of the methods actually make the expanded tissue very fragile. However, some of the more recent ones, specifically Magnify and Pan-EXM, increase stability quite substantially. So in the expansion microscopy that I worked with during college, you pick it up and it basically falls apart in your hands if you hold it wrong. But these newer ones like Magnify and Pan-EXM are able to make it so that it's almost like a piece of glass or a piece of plastic. I mean, it isn't actually that material, but it like feels that way in terms of stability. So at least from what I've heard from people, I haven't handled those myself particularly, but I hope to at some point. So this is expansion microscopy. What if we combined expansion microscopy and X-ray microscopy so that you could take advantage of the speed of synchrotrons while imaging the higher volume of brain tissue from the enlarged, expanded samples? But there remain challenges, engineering challenges that can, in my opinion, be overcome. So expanding cubically dilutes the signal, of course, because it's making everything bigger in all the directions. And what I mentioned earlier was that there was a recently published preprint that may be a solution to this. So when I was an undergrad, I was running into this problem where I didn't see anything because when I stained the tissue and expanded it, it basically lost it lost the density of signal enough that the X-rays could be absorbed. I was using a laboratory X-ray microscope at the time, so it was slower, but it was similar principle. However, unclearing microscopy, which is a method by which essentially you're able to fill in the gaps after expansion, was recently put out in a preprint by Masad et al. Ons is her first name. And essentially what's done is that you link horseradish peroxidase to all the membranes as well as all the proteins in the sample. And then you do a silver stain. And the silver stain, you can also do diaminobenzidine, by the way. Um, Basically, you do a stain, and this enzyme catalyzes the reduction of the silver ions, and this deposits visible, visibly dense, basically replacements for the part of the tissue that was diluted out when it was expanded. And for this reason, you can actually see cells, literal like single cells that are. This was expanded 20x approximately or 25x. You can literally see the cells visible to the naked eye when this is done. And when I saw this, I was like, "Oh my gosh, this is amazing! This could solve the problem that I had." And there's also images of this under the microscope where you can see the membranes and the boundaries of organelles. It's really great. And so I think that this would solve the problem. Um, However, there's a second engineering problem, which is that synchrotron X-rays are so bright that they can damage samples, even frozen hydrated samples. Um, and then from there, the recent expansion recipes are relatively fragile. Even the recent expansion recipes, which are more stable than the original ones, they're still relatively fragile. Although if you put it under cryogenic conditions, it might not matter that much between the two. But essentially, I think there needs to be some research done into, at least for large scale stuff, post expansion stabilization. So rapid cross-linking of some form after expansion, obviously rapid freezing, and then of course sectioning so that the radiation dose is less overall. I'm thinking of maybe 3.3 by 3.3 by 100 centimeter cubed scale blocks. Oh, that shouldn't be cubed. That should be just centimeters. And this would essentially be columns, and then you could image at the along the z-axis. You could image and not expose the rest of the z-axis to much radiation. So that's what I'm thinking, and I'm actually currently working with a group at EMBL who they say that they're planning on doing some of these experiments once they're able to hire a postdoc, and they want to try it out. And so I'm optimistic about that, but they do have some teaching obligations that they have to take care of first. And so they're currently putting it on hold. So in the meantime, I'm always open to other collaborations. And of course, I would need to communicate with them about that. But I'm definitely interested in ways of getting this to move forward. Um, so here's just the proposed workflow. You would start with a brain. This is just a nice image of human brain that I made. You'd expand it probably at least 11 times, if not 20 times, rather than 4 times, as is shown here. You'd use the unclearing method that was developed by Ans Masad, and then you'd stabilize it using some as of yet not invented way of cross-linking post-expanded tissue. Then you would section it into large columnar chunks, and then you would image the chunks and reconstruct the brain. And down here is just like what it would look like at the single neuron level. Okay, so this comes to the big question, which is, are synchrotrons really fast enough? So there is a mesoscale connectomics, so not nanoscale, not enough for not enough for whole brain emulation, but there is a mesoscale connectomics initiative called Synapse, the Synapse Consortium, which is actually based in Asia, where they have been able to image at low resolution mouse brains using projection times of 1 millisecond. As was mentioned earlier, a projection is just the 2D image that gets taken at different angles for the reconstruction of the tomogram. So if a projection takes 1 millisecond and you have to do a certain number of projections, at least n times pi over 2 projections, where n is the number of pixels along the x-axis, then it'll take however much time is 1 millisecond times that number of projections to reconstruct a tomogram. Basically, the conclusion that I've come to is that synchrotrons do have the potential to image the whole human brain in 1 year or less, but you need much larger detectors to do this. So you need to develop a custom beamline with a large detector of at least 18.6 millimeters by 18.6 millimeters to be able to image enough material, enough volume at a time to reconstruct an 11-fold expanded brain. And that would give you 27 nanometer effective pixel size after expansion, 11-fold. I have a whole MATLAB script where I did all these calculations. I am optimistic that larger detectors can be made because there's been some efforts at making multiple millimeter detectors with reasonably small pixel sizes. We're aiming for 300 nanometer pixel size, but it's likely, and I haven't seen any that are able to reach that while still having enough pixels to cover 18.6 millimeters. But there was a recent announcement or about a year ago, of a 560 nanometer pixel size smartphone camera, interestingly enough, with 200 million pixels, which essentially, if that was a square camera detector, it would be about 14,000 by 14,000, 560 nanometer pixels, which I believe came out to a few millimeters. And so there's definitely, because you would need 62,000 300 nanometer pixels for a 18.6 millimeter on a side detector. That's the number of pixels across the side. And this is the number of projections you'd need by Crowther's limit. A single tomogram using this system would take about 100 seconds to acquire. Anyway, the message is that I'm optimistic that you can potentially develop a detector large enough to do this. And interestingly enough, a mouse brain you could do in 1 month with substantially less restrictive parameters for the engineering. A 300 nanometer pixel size could be achieved with a 4x objective lens in that case. The reason an objective lens couldn't be used for the human brain version is because the objective lenses have certain engineering limitations on them as well that would make it very difficult for an objective lens to cover 18.6 millimeters of because of its numerical aperture. But again, I'm getting into the weeds there a little bit. Okay, so the point is, yes, synchrotrons are fast enough, but we need bigger detectors. And then for the point of, to further support this, I did some calculations using a model from Du et al., which was a paper that discussed not expansion microscopy, but just straight up X-ray microscopy as a potential way of mapping the brain. And in that paper, they basically have a set of calculations that they use to determine how many photons are needed to image a certain sized feature. And this is the number of photons per pixel. I found that you would need 10 to the 14th photons in total for the tomogram that I described of 18.6 by 18.6 millimeters. If you divide by the area in millimeters, this would be 2.83 times 10 to the 11th photons per millimeter squared, which I actually found would lead to 3.19 tomograms per second, which is probably a substantial overestimate. That makes sense because this is theoretical and it doesn't account for the real world issues that one would encounter. So as opposed to my previous calculation, which was 100 seconds per tomogram, this would be more like 0.3 seconds per tomogram. But yeah, I mean, the point is that this further supports the idea that it is definitely possible with modern synchrotron beamlines to reach fluxes of photons that are enough to map the human brain rapidly, provided you have large detectors I mentioned. All right, we're almost done here, and then we'll get to discussion. Radiation dose is the last thing I want to talk about. There's also, based on Du et al.'s model, a equation for radiation dose. I calculated about 2 times 10 to the 10th gray, which is a unit of measurement that is equivalent to 1 joule per kilogram of energy transferred by the X-rays, and that's the radiation dose. So 2 times 10 to the 10th gray per pixel is the amount of radiation dose that you would need it at the flux that I stated previously. And the interesting thing is that this is too high, honestly, for keeping the tissue together. However, at least based on what Du et al. Were describing, they were saying basically the maximum that you should be tolerating is closer to 10 to the 9th, whereas at 10. But however, I do want to say that tissues in other experimental contexts have tolerated actually similar doses to 10 to the 10th gray. particularly frozen hydrated single cells have been studied this way, and so it's not like it's completely out of the question, but stabilization techniques, as I described earlier, will probably be necessary to sort of re-embed the expanded tissue in something that's stronger so that it doesn't fall apart under this level of radiation. All right. Oh, one more thing. Sorry. So a how much would all this cost? So new synchrotron beamline typically costs around $10 million. So you'd need to build a new beamline into this facility. Most of that would probably be construction costs. But of course, initial smaller scale experiments might generate the data that shows that this investment is worthwhile. Because if we really can show that all the stuff that I've described is possible, I think that people would be willing, at least hopefully, would be willing to invest enough grant money or whatever, wherever it may come from, into building a dedicated connectomics beamline that could image a human brain per year or something along those lines. But yeah, it definitely is something that obviously there needs to be a lot of preliminary data around, and that's what I'm trying to do with the EMBL folks. All right. So that presentation went on a little longer than I expected, but I'm now open to questions and discussion. All right. Thank you, everyone.
Wonderful. Thank you, Logan, for that fascinating presentation. And we do have a number of questions from our viewers. The first one I will pose is from Alan Crowley, who wonders, are synchrotrons non-destructive, and could one be large enough to put an entire brain in? Well, a synchrotron is a huge building, so likely multiple living human brains fit into it at present. But I think he was asking the question, can microscopy using the synchrotron essentially encompass all of the connections and processes within the human brain for analysis?
Sure. So to answer your question, synchrotrons are relatively, and X-ray microscopy in general is relatively non-destructive. With the exception of the radiation issue that I mentioned, if you have enough, if your synchrotron is bright enough, it may start to degrade the tissue. And in the case of the speed that you would need, it probably would be a question as to whether you could stabilize the tissue enough to prevent it from being degraded. However, generally speaking, yes, X-ray microscopy and synchrotron X-ray microscopy are a non-destructive imaging modality. As to the particularly with propagation-based phase contrast synchrotron X-ray microscopy, you can have long sample to detector and sample to source distances, and it can actually be beneficial, as I understand, to have those longer distances. So it definitely is possible to have very large samples in the system. There is some fall off of penetration of the X-rays as you get deeper into very thick samples. So it would probably be wise, if you wanted to maintain resolution and image quality, to section into, like I said, sort of those columnar French fry looking sized objects. But yes, it is hypothetically possible to put an entire brain into a synchrotron sample chamber, because I know this because people have actually put entire lungs in and imaged it relatively low resolution. So there was a paper in forget what journal. It was one of the Nature journals, I think, maybe Nature Methods, where they were able to image coronavirus infected patient post-mortem lungs, as well as a bunch of other organs, like I think, I don't know, gallbladder or something weird like that, as well as brain, but it was too low of resolution to be useful for connectomics. And they did image entire samples that way. At I think it was the smallest sub region that they imaged was I believe it was their voxel size was something on the order of like 2 micrometers. So obviously much bigger than 0.3 micrometers, which would be the 300 nanometers that I mentioned would be necessary, but still shows that the technology is advancing. The other cool thing is that synchrotrons are just always getting better. There's been what's been called 4th generation synchrotrons that have been coming out lately, which are supposedly much faster than the 3rd generation ones. That was actually what was used for this particular study I mentioned with the lungs. However, I didn't really see where the big speed up was in the lung study, but I think they weren't maybe taking advantage of the full potential of it in that case, because they had different scientific questions they were trying to answer, and as well as limited funding, and they were just using a pre-existing beamline rather than designing a beamline specifically for that purpose. So anyway, there's some nuance to it, But the point is, yes, you could put an entire brain in, and imaging large samples is feasible.
Mhm. Yes, thank you, Logan, for that question. And there is a related question from Alan Crowley. Can synchrotron X-ray scanning tunneling microscopes resolve surfaces down to single atoms?
Okay, yes. So I believe this is a little outside of my area, but I do know that uh, I'm actually not super familiar with X-ray scanning tunneling microscopes, but I would assume that they work similarly to other scanning tunneling microscopes. Maybe you're thinking of scanning transmission X-ray microscopes. I'm not sure, but anyway, there are various types of X-ray microscopy in that can be used with synchrotrons. One of them is scanning transmission X-ray microscopy, where it's slower because you have to scan a focused point across your projection image. However, it is much higher resolution, so you can get down to nanometer scale, and even sub-nanometer scale, I believe. As to single atoms, I do know there was a recently published paper that's been actually kind of all over the news lately about an X-ray. I don't know if it was specifically a scanning tunneling or if it was scanning X-ray transmission. I haven't had a chance to read it yet, but there definitely was images of single atoms that were taken using this microscope, and it was a big headline. Actually, I'm not sure if maybe you've heard of that, and that's where this question's coming from. But it was a breakthrough. There's also ways of reconstructing individual proteins. It's an alternative way. Synchrotron X-ray tools, not microscopy, but synchrotron X-ray tools can be used to reconstruct the atomic structure of proteins as well. And I know that that's sort of a whole other field that's like adjacent to this one. Um, does that answer your question?
Well, I think Ellen in the chat will indicate if it did. I think it's quite a helpful clarification. I do want to ask a question of my own on expansion microscopy, and this was discussed a bit in our YouTube chat as well. Essentially, the concern from some researchers and some transhumanists who want to preserve the human brain and ideally the individual human being through a kind of whole brain emulation type process is that expansion microscopy would be a destructive process because you are physically expanding the cells, as you mentioned. It can be an expansion, say 11-fold, and there's an advantage to that because you magnify the particular components of the cells, making them easier to study. But is there perhaps a possibility to use expansion microscopy with a sampling approach? So instead of taking the whole organism or the whole organ, you take individual cells from it, you study their structure, their contents, and then you extrapolate from that somehow to create an emulation of the organ, including the brain.
Sure, sure. So, yeah, that's a very interesting question. And for those motivated by longevity questions, including myself, I mean, I do want to live a very long time, if not forever. Um, but for me, I've always sort of set the longevity thing as sort of a secondary priority relative to the preservation, continuity, and continued growth of the human species. However, I do think longevity is very important, and so I will mention that I do think that there are some possibilities here. So one possibility is Ken Hayworth and Randall Cohn have made some interesting philosophical arguments that I'll admit I haven't fully wrapped my head around yet. That not that the destructive brain scanning is still compatible with certain ideas of consciousness being preserved. I don't know if I fully agree with those, but I think that it's an interesting area to explore, and they've published a few papers on it. Um, but as to something that I personally think is more directly feasible, uh, the if you want to image a human brain and still maintain sort of that continuity. One idea I would have would be there are people who have been able to survive with only one half of their brain. And so I would say, okay, what if there was some surgery where you could remove a hemisphere, um, image it, and then like have the person, I don't know, in in under the whole time, like and then like preserved or something. This is a further future, of course. Image have the person um, just basically remove one of their hemispheres, and then image it, computationally reconstruct it, use the data from that reconstruction to create a electronic or some kind of other neuromorphic electronic replica of the hemisphere, put that hemisphere back in the person, wake them up for a minute so that like connectivity and information can start to transfer between the hemispheres, then put them under again, remove the other hemisphere, and do the same thing. And then you have them basically transferred into a electronic version of themselves. Of course, they'd still be in their body, so you'd want to like then take the two electronic thingies out and like put them inside of a larger system or something, which may not be exactly what people always envision when they think of mind uploading, but I think it's maybe most the most feasible route given the constraints that we currently have. Um, so yeah, that would be my advice if you want to live forever this way.
Very interesting response. Thank you, Logan. And by the way, for anybody who is interested in the philosophical arguments that Kenneth Hayworth made, we had a virtual Enlightenment Salon with him and Dr. Robert McIntyre on September 22nd, 2020. And this was one of our longest virtual Enlightenment Salons, over 3 hours and 47 minutes, discussing both the science and the philosophical implications. And I have some disagreements with them in regard to destructive mind uploading. I think frequent viewers of the virtual Enlightenment Salons know my position about the necessity of the continuity of physical processes within the body to preserve the self, or what I would call the I-ness. And we have an interesting comment as well from T. Theodorus Ibrahim, who writes that he would suggest leaning heavily on computational deduction of connectomic structure, allowing us to relax the constraints on acquiring very high quality imaging data. So very interesting perspective there. And now let us go to Dr. Jose Cordero, who has several questions for Logan. Please proceed.
Yes, hello, Logan. First of all, I want to congratulate you for such a fantastic and comprehensive presentation. I really loved it, and it shows that you are working on so many fascinating areas. So my sincere and deep congratulations. I have 3 questions. Let me begin with one, and then later maybe second and third questions. I actually wrote a book, which is a bestseller in Spanish, La Muerte de la Muerte, and in other languages, and it's coming out in English now, The Death of Death, published by Springer Nature. And I do a chronology of the future. I go all the way to the year 2099, and I use many of the numbers by my friend Ray Kurzweil. But I also use some other forecasts, including predictions by Elon Musk and British Telecom and some other people and companies that talk about the future. But my book, basically, The Death of Death, is about death, killing death. And I see 2 ways of killing death. The first one is hardware immortality, and the other one is software immortality. Hardware immortality basically is biological immortality as defended by Aubrey de Grey. And software immortality is computational immortality as defended by Ray Kurzweil, both of whom are my friends and both of whom write on my book. So my question to you is that both of them more or less agree that we will reach immortality by the year 2045, both biologically and computationally. So my question to you is, what is your take on that and on the 2 different types of immortality, both by 2045?
All right. So I definitely, I'm definitely not necessarily thinking that 2045 is the best estimate personally. I do think it'll happen eventually, but I'm a little bit more cautious about making those nearer term kind of projections. So with regards to hardware and software immortality, I'll answer that part first. I think both are very worthwhile, and I think both will sort of come about in synergy. Probably the the Aubrey de Grey style first will come, and then there will be sort of an escape, what's people have referred to as longevity escape velocity, where people will be living longer and longer, and then eventually they'll live long enough that that we reach a point in technology where we can make much more radical advances. So my thinking is, okay, I don't personally, from a scientific point of view, I just don't personally think it's very feasible to have mind uploading by 2045. I used to think that actually, and I'd love to think that if I could, but just all the stuff that I've read, all the stuff that I've experienced since then, as a scientist, as a futurist, as a writer, as life. Unfortunately, I think that it's quite a bit further out. I don't want to say anything super specific, but I'm thinking in the range of 150 to 200 years from now, that like mind uploading style immortality could be possible. However, I could be very wrong. I mean, you can never know with these things. Maybe the advancements in AI that I'm not as in that field, maybe that will greatly accelerate this type of research in a way that I haven't accounted for. So maybe I'm wrong. Maybe it'll happen by 2045, and I'd love that if it would. But I personally, as a biologist, I'm a little more cautious just because the incredible complexity of biology. I think that everything is accelerating, but I think that the problems that are needed to be tackled are so challenging that even accelerating progress will take a little while to address them. However, I do think that we do have the potential of everyone here has the potential to experience this type of immortality. Not the guarantee, of course, but the potential. Because I think Aubrey de Grey's style, which is more biological longevity, gene therapy, pharmaceuticals, etc., that could extend our lifespans by 20, 30, 40 years, would put many of us at the point where we could reach that sort of longevity escape velocity. And then let's say mind uploading does take 200 or 300 years from now, many of us may still be around by then. So that's my personal take on it. And I know a lot of people here might disagree, but I think that I'm coming very much from a scientist's point of view, and perhaps I'm too like embedded in the details of the laboratory. But I am a little more cautious about saying it'll happen that soon.
Okay, so let me go to the second question, because Ray Kurzweil just sent me the manuscript of his coming book next year, The Singularity is Nearer.
Nice. Awesome.
And obviously, he defends the 2 dates that he has been talking about for almost 4 decades. By 2029, we will pass the Alan Turing test. And also, according to him, if we make it to 2029 to 2030, we will reach longevity escape velocity and basically live long enough to live forever. Still aging until 2045, when we reach the singularity and immortality through biological rejuvenation. Okay, those are the predictions by Ray Kurzweil that are going to come out next year in his new book, and that he has been forecasting, for decades, and he has been 83% correct in his predictions. 83%, I find that absolutely incredible. In any event, he's older than me and you, so he's a bit more worried, but he's following all these exponential developments, and he thinks that we will reach longevity escape velocity that soon. It sounds quite early, even to me, but let's hope what happens. In any event, in terms of computational immortality, now we have Elon Musk and as you know, Neuralink and many other companies that will be able to basically read some of our brains, part of our brain. So we might not need to read it your way. We might read it in a different way. So what do you think about that? Also with artificial intelligence, that no one thought it was moving as fast as it is moving now.
Yeah. So I have great respect for Ray Kurzweil, of course. I sort of grew up reading his stuff, and I've always found it super fascinating and inspiring. With regards to Neuralink and brain-computer interface technology, I think that is very promising for a variety of reasons. My thinking on that is that there may be a way of almost like remotely diffusing consciousness, and of course this is very speculative, but there's sort of a possibility that you might be able to, particularly if there's a way of combining gene therapy with or genetic interventions of some kind with optogenetics and as well as external devices, there might be a way of almost like making one's consciousness sort of an exocortex style. And there may be a way to, I don't know. I mean, I'm a little hesitant to say that that would actually facilitate something like mind uploading, particularly since you wouldn't be able to, you wouldn't necessarily be replacing anything. You'd just be reading from reading and writing into the brain. But there may be some possibilities there. Also, the scale needed to read and write at the level that would be needed for mind uploading, I think, is extremely high relative to what we have now. That said, I think all these technologies are super promising, particularly ones which involve, particularly the ones which are. Oh, there was one that I was hearing about recently that sounded really promising, but it's escaping my memory right now. It was not Neuralink; it was a different one actually. But yeah, there's. I've had some ideas about actually combining optogenetics and upconversion nanoparticles, which are a type of nanoparticle that converts infrared light into shorter wavelengths like blue light, and they're essentially a way of making optogenetic tools less invasive. So you would hypothetically be able to inject a package, a nanoscale package carrying the DNA necessary to express the ion channels that respond to blue light, as well as upconversion nanoparticles. They would travel through the blood-brain barrier through methods that I can talk about later if you're curious. I have ideas for that. And then they would enter the brain and enter the neurons, and then you'd essentially have each neuron having a little particle in it, or more than one maybe, as well as the DNA expressed in the ion channels. And then you would be able to stimulate specific regions of the brain, or even maybe specific neurons, depending on how focused you can get your infrared light. And you would be able to write into the brain in a non-invasive way. As to reading the brain, I know there have been some ways of using ultrasound, which are promising for having like almost like crystals that respond to ultrasound, and then when the electrical activity of the neurons happens, the crystals change their frequency of the bounce back from the ultrasound, and I know that has potential for being pretty high resolution writing of things into the brain, or reading of things from the brain, as well as writing, because you can also stimulate that way or in a similar way. But yeah, I think there's some possibilities for brain-computer interfaces, and I do look forward to a world where we can slowly become more and more interconnected with technology in a fashion that allows us to interact with technology and our environment in a more fluid and dynamic fashion. And I think that will accelerate everything else. I'm not sure if mind uploading will necessarily happen that way, but again, I think it's not something we can rule out. It's just something that may not be the most direct route or the most likely route.
Okay, and my third question, by the way, do you have an immortal jellyfish in your background? You have something in your back? And
oh yeah, those. I don't think any of those are that. I'm actually not sure if it's any of those are that particular type of jellyfish, but I do have a poster on my wall I got at a local store of several different species of jellyfish. My place is a little bit sparsely decorated at the moment, but that was one thing I wanted to put up because I thought it looked so cool.
Beautiful, beautiful. So my last question is about cryonics. Also, because we know Kenneth Hayward, Randall Kuhne, McIntyre, and they are not too convinced about cryonics, but there are other methods of biostasis. So biostasis in general. So what is your take on cryonics? Also, because there are some cryonics people watching this. Like I saw my friend Rudy Hoffman from Florida, and he's very interested in cryonics, and so am I as Plan B. Plan A is immortality. but plan B is some sort of biostasis, and the most used now is cryonics. So what is your take on that?
My take on cryonics is that it's really underappreciated at the moment. I think it's something that is, uh, I think it's something that is a real science, but some people sort of categorize it as a pseudoscience, including Wikipedia, interestingly. And I don't really think that's entirely fair because it's just something that hasn't received enough attention to really progress in the way that it should. I think it's a worthwhile endeavor. Obviously, there's a lot of problems in the world, and people have made the argument that, if you put too much stuff in one problem that may seem like I don't know that it'll take away from others, but I don't really believe in that. And so I think that there's a lot of smart people out there who are working on valuable problems. Cryonics may not be my first priority, but it's something that I think is worthwhile to have around as an option for people. And I think that it's something that may be useful in the future for things beyond just longevity. It could be useful for things like deep space travel. And so I would love to see more research done in cryonics and brain preservation in general and body preservation and so on, because I think it's something that could be quite valuable. And I just don't think there's been enough done with it. I mean, I know Robert has done some excellent work with perfusion of the brain right as people are passing, and I think that that's probably the most promising technical solution so far with regards to cryonics style stuff. But I would love to see more investment in that area because it seems like something that just because it's weird, people think that it's not worth trying. And because it's sort of it's just something that society has sort of categorized as not worth doing for societal reasons that I don't really personally agree with. And so I like cryonics. It's not my first priority, but I think it's something that is worthwhile doing, and I'd love to see more put into it.
Thank you. Thank you.
Thank you very much, Logan, for your responses to Jose's questions. And there is a bit of feedback from our audience members regarding this exchange. So John H. writes, old people hope that things happen fast because we feel ourselves running out of time. And well, to some extent, I feel myself running out of time as well, because I would have hoped that the preceding 19 years during which I have been involved in the life extension movement had generated more progress than that. And there are some societal, cultural, political factors that led progress to be slower than many of us expected at the time. Mike Lazine writes, he hopes that Ray Kurzweil is right, so that by the time he is 70 years old, the technology should be affordable to masses for longevity tech. Now, John H. also expresses a view that I think you would identify with as well. We literally don't even know what consciousness is, how to quantify it or isolate it, or to transmit it out of our bodies and brains. And this, of course, is both a philosophical question as well as a neuroscience question. And I'm curious as to your thoughts on this, Logan, with regard to whole brain emulation, for instance, particularly in a different medium. To what extent would that preserve, in your view, the original individual's vantage point upon the world? So that if one transitions from one's current biological substrate into this new substrate, will the experience of that person be preserved? And what would you hypothesize the prerequisites of preserving that experience would be? Since it seems this entire discussion is premised on there being some sort of analogous structures to those of the biological human brain, which would still enable self-awareness to continue.
Sure. So I guess that kind of depends on whether you're accepting the copy problem or not. So there's sort of two answers to this. The first is yes, the consciousness issue is something, actually three answers. So to address the first part, consciousness, we don't really know what consciousness is exactly. I suspect that a panpsychist view or something akin to it may be a explanation for consciousness, or at least a mechanistic way of understanding consciousness that could work. But I don't know that for sure. I do think that it may be possible to gather evidence for the idea that consciousness could be transferred between different substrates by actually using neural prostheses. So it's almost like an experiment. So you put a neural prosthesis into someone that replaces part of their brain. What is it like? And they can actually report to you hypothetically in this situation. And there are some very, very early neural prostheses that I wouldn't really classify as being invasive enough for this particular thing that have been developed. Like Theodore Berger and Dong Song have developed a hippocampal prosthesis, but it's really just stimulating and receiving from part of the hippocampus. So I don't know if I would necessarily say it could perform this type of experiment in a reliable fashion. However, I do think more are coming, and I think that as we learn more about how neural prostheses interact with the brain, we'll learn more about what consciousness can do. Maybe not what consciousness is, but how consciousness can move around. With regards to the main question, the main question I believe was that, is it like how much detail would you need to recreate the person, either as a copy or as a mind upload that is maintained with continuity? I'll talk about the just assuming that we're wanting to create immortal copies of ourselves so that they can go populate outer space or something, and actually preserve the individual. I would say that you would need. I mean, for one thing, you would need the morphology of the neurons. You would need the connectivity, but you would also need labeling information. So you would need to label at least 10 or so, maybe more, proteins, transcripts, etc. Because you would also need to quantify things like gap junctions. You would need to quantify neuromodulators. You'd need to be able to, or at the very least, infer these things. So that should come with that caveat. You'd be able to quantify things like gap junctions. You'd be able to quantify things like cell type. And one thing that I think is super important is finding a way to translate from morphology and neural context to cell type in the sense of its genetic expression patterns, so that we can infer its spiking patterns and its electrophysiology in general. So things like ion channels, of course, various types of ion channels, especially on the dendrites. There's a variety of computation that happens there that may be important for individuals. Even certain transcripts within the synaptic boutons and the dendritic spines may be important. So there are some things that would need to be labeled for sure. X-ray microscopy is something that could potentially develop to the point where that would be possible eventually. Right now, it is possible to do labeling in X-ray microscopy at a more primitive level than it is with fluorescence microscopy, because you can do like multicolor, like maybe 2 or 3 colors. But there might also be some other ways of extending that further in the future that I would like to look into more. But I do think that it'll evolve over time where multiple imaging modalities are used, at least for in the beginning. Like the first time we map a brain and try to create an emulation, there will be multiple imaging modalities involved, and then maybe eventually it'll transition into one thing or the other. But you would need to definitely understand more than just the morphology and the connectivity. And then just like getting the emulation to work, you'll need to do a lot of benchmarking. You'll need to do a lot of testing along the way of smaller parts. You'll need to make sure that you understand, because you can't just take a bunch of data, put it in, and run the simulation and expect it to work the first time. You'll need to do a lot of validation along the way. And even once you reach the point where you're actually creating the emulation, there might need to be a fair amount of tweaking and so on and so forth. A lot of it's very speculative, of course, and so I'm keeping it very open here. But that's my thinking with regards to the level of detail that would be needed for whole brain emulation. For mind uploading, I guess I'll go back. It's sort of the same deal, but I'll go back to my suggestion of hemispherectomy followed by replacing with a synthetic hemisphere, and then doing the same with the second hemisphere. So there you go. That's my answer.
All right. Thank you very much, Logan. And we have a few more comments as well on the subjects that have been discussed. Rudy Hoffman writes that cryonics is currently in the proto-science rather than pseudoscience stage, and he believes your observations, Logan, are spot on. He also notes that Ray Kurzweil is actually signed up for cryopreservation. He's just not as vocal about it as someone like Aubrey de Grey would be. But Aubrey de Grey, who, of course, is a proponent of achieving biological longevity, escape velocity, is also funding a startup in cryonics called Kynice, which is founded by Tanya Jones and is studying a new cryopreservation technique called persiflation, which uses gases to essentially preserve tissues, hopefully with greater fidelity than vitrification. And the Longevity Escape Velocity Foundation, which was recently founded by Dr. Aubrey de Grey, donated $1 million to Kynice in April of this year. So hopefully some significant advances in studying this technique will be made. And I think it's really important to also consider some of the possible synergies between cryonics and whole brain emulation, particularly because if there can be more reliable cryopreservation techniques for human bodies and brains of patients who become deceased in the near future, then this could buy time for advances in brain imaging, especially non-destructive brain imaging, that could allow many decades later for these patients' brains to be more faithfully emulated.
Absolutely. Thank you.
Yes, yes. Now let's go to Ben Ballweg for any questions or comments from him.
Thanks for being here today, Logan. You had said something about your longer timeline perspective, and you kind of hinted at that just the biology of things makes you think that we're not going to do things as fast as maybe the USTP would like. And then I did hear Gennady said something about like societal, maybe societal issues that slow progress a little bit, that we just aren't as organized, or we're not organized in the right way as we could to be able to speed some of these technologies. I guess I kind of wanted to hear your perspective on what could we change. I'll say like the biology, those limitations are immutable, but what kinds of societal adjustments could be made to make you a quicker than your current timeline perspective?
Yeah, so I think that there are several things that could be done. Particularly, there are some encouraging ideas of there are some encouraging ideas of alternative funding that may be able to accelerate science that have come out recently. So Adam Marblestone and his organization Convergent Research are currently trying to get philanthropic funding for organizations called FROs, focused research organizations, which tackle particular research problems in a more organized fashion that may not be fall into the traditional startup or academic project categories. So being bigger and more challenging than most academic projects, but not necessarily having a direct return on investment, like at least in the near term, like many startups. And so organizations like Convergent Research, which are funding FROs, a lot of the FROs that have come out of Convergent Research seem very promising. One of them is working on light sheet approaches to connectomics, which, as mentioned in my presentation, are potentially responsible for substantial advancements relative to the calculations that I did. We'll see how well that pans out. It would still take a lot of investment to map a single human brain in their methodology, but it's something that they would be. It may be the first group to do it. I don't know. Maybe the synchrotron stuff will be the first, but you never know. I mean, it's interesting anyway. So FROs, alternative funding organizations, restructuring of the funding systems in general, because right now, I mean, as much as it's interesting and fun to write grants, the grant system is a little chaotic overall, and there's an increasing recognition of the need for scientific funding to be both increased and better organized. And so I think that could help. Commercial sector stuff could definitely help as well, particularly advancements in AI being applied to biological problems. I think may be interesting to see what happens over the coming years. So one thing is resolution of connectomics, for example, and this is just one example among many. If you could image at a slightly lower resolution, it would greatly increase the speed. So, like, let's say you only needed 50 nanometer voxels. However, a lot of the information is going to be lost then, and so if you if there's some way of creating an AI that can infer that information in a relatively accurate fashion, in sort of like almost like a generative AI that fills in the missing pieces or upscales the resolution. And I personally, I don't really know whether this is actually feasible or not, so I'm sort of just speculating here. But I think that that type of AI, like applied to problems like that, well as many other biological problems, such as how does the body work with this big network of genes and gene expression and proteins and peptides and glycogen and with sugars flying around, all this different stuff. How does all work as a network? AI might be able to start to integrate a lot of that information in such a way as to make more sense of biology. And I would be curious. I mean, I don't know yet whether it'll reach that point in the next 10 years or 20 years or whatever, where we can really say, okay, you train this AI to do whatever, and then now you can predict exactly what will happen in the body if you perturb X, Y, and Z. But that would be something that if advances in AI do that, it might be super helpful. I see a comment about quantum computing. Honestly, I'm probably not the person to ask about this one, but if quantum computing does become a thing, particularly if it accelerates computational chemistry in such a way as to run fairly complex simulations in parallel, I think that there might be some very interesting outcomes that could be useful for making biology more engineerable. So again, all of this stuff is in the context of these problems are still very hard. Biology is just super messy and super complicated, and I think some people tend to underestimate just how difficult it will be to figure it all out. And I would say I'm one of the more optimistic people in the world about this, but maybe not as optimistic as Ray or Aubrey, but still more optimistic than most. But I do think that all these things will be helpful. I think that there's definitely possibilities for it going faster than I expect, but I wouldn't necessarily say it's for sure. But yeah, I think these different factors could help. But then, so far as society goes, in a broader sense, like societal opinion, that's actually something I am super concerned about that, because I feel like currently we're in this stage where just everyone is, well, not everyone, like the vast majority of people don't really understand science, they don't support science, and they certainly don't support transhumanist goals. And some people even seem to support like almost like the reverse, like human extinction, which honestly I think is more of a coping mechanism that people employ because they think that it somehow makes them like special to like hate humans or something. But I don't know. That's just my personal pet peeve when people go on about that. But yeah, I do think that societally we have reached a point where um, we're starting to mature as a society and we're having growing pains, and we need to find our way to a more optimistic mindset if we want to make it as a species. And I think that it's possible. I believe what was his name? The fellow who writes about history as a mathematical construct. I'm blanking on his name right now, but he's talked about oscillations, and he's discussed essentially the idea of there being these oscillatory periods in history where, particularly in the context of power, where certain powers will rise and then they'll be overthrown by the by another power eventually. But I think that the oscillations idea can apply to mimetics as well, to the idea of like how certain ideas rise in society and then eventually fall. And so obviously I wasn't alive during the 60s or whatever, but I know I've talked to some people who were alive during those types of time periods when there's a lot of strife and a lot of transition, a lot of sort of I don't know, just a lot of conflict, a lot of younger people having sort of a disillusionment with the system, which, to be fair, the system is kind of messed up. But I mean, there's other ways of approaching it than the ways that that those sorts of people approach it. But anyway, yeah, I think that we do have a hope of reaching a point where some of this stuff eventually calms down. Um, a lot of the a lot of it may come from as we get better ways of transmitting information, and better ways of regulating the information without being authoritarian, ways of channeling information such that it doesn't. I mean, I guess I'm talking mainly about social media here. Ways of sort of managing the fact that we are all interconnected in better ways may come about in the next 10, 15 years. And if we can do that, I would hope that society would reach a point where we're more optimistic, as well as new generations coming about, people growing up in different times. Maybe in 10 years things will be a little bit better, and people being born then won't even remember all the stuff that people think is wrong now. Personally, I've looked at the data, and now is still the best time to be alive, in my opinion. But just on average, but not necessarily for every single person. But I think that there's a lot of perception because of the way that information is transmitted in society that it's actually the worst time to be alive, which is definitely not true. And so, yeah, I guess it's just an information problem. It's something that I think it's a maturity of the society problem. Our civilization has some growing pains, and my hope is that over time it'll get better. I don't know if it will, but I hope it will, and I am optimistic.
Yes, thank you for that response, Logan. And I think this is an excellent bridge into a conversation on science fiction. So you have a post entitled Rational Romanticism on your website, which I would encourage people to read. And an excerpt from this post, I think, aligns well with what has been discussed. You write, if we cultivate rational romanticism, we can achieve wonders. The cultures of imagination and science are currently opposed, but by bringing them together, science will not be limited by status quo bias or stifling pragmatism. Likewise, art will not be limited by an aversion to the beauty found in science and technology. In this scenario, science and art will be synonymous. Emotional intensity will be channeled towards solving challenges and enriching the human experience. With the combined power of science and emotion, we can make dreams into reality. So if you could perhaps elaborate a bit more on what you consider to be an effective role in science fiction in terms of transforming the culture, transforming people's perceptions of what is possible, and how science can further improve our lives and the quality of the world around us.
Absolutely. So, yeah, I as mentioned, am both a reader and a writer of science fiction, and it's something that I really love to. I really love just as a craft. But on a broader sense, science fiction can play a pretty big role. I think. I mean, a lot of the ideas that have been discussed today, gene therapy, mind uploading, whole brain emulation, etc., have been very much rooted in science fiction. And without people being imaginative, without people having visions and wanting to create something, seeing these possibilities. I mean, even things like space travel has been a big part of science fiction. And to some degree, there's an interplay between does the society stimulate the fiction, or does the fiction stimulate the society? But I do think that the fiction, in many ways, does affect society. And right now, the most popular science fiction is mostly pretty dystopian, which is kind of a problem, a big problem, in my opinion. And I would like to see something more akin to what was happened in the, I guess, the 50s through maybe the late 60s, or I think even the 40s, maybe to some degree. I don't remember the exact timeline that people say it is, but the golden age of science fiction, where writers like Arthur C. Clarke and Isaac Asimov were really exploring these deep questions of science and spirituality and the future and life on other worlds and robotics and what it means to be alive, and as well as just looking at the universe through this lens of intense wonder. And I do see value in the types of science fiction that take a look at challenges, that take a look at that aren't like entirely utopian, but they acknowledge that there are some challenges remaining, and I think that that's very important. But the attitude is also very important. The way that they do that, and right now it feels to me like a lot of the science fiction is almost lazy. And to some degree, that's the fault of a lot of it being in film and a lot of the film stuff being very grabby, like the stuff that makes the most money in the film being grabby and making people afraid because fear can be a strong motivator for people. But I would really like to see a way of, I mean, maybe even there would be a way of people founding some kind of, and maybe I'll work on this at some point, founding some kind of a group. And I think some people have worked on this to some degree, but it just hasn't really exploded yet. But almost like a science fiction for making society better foundation. So, and something that would be fairly large and well organized, and transparent as well, so that people don't think it's just propaganda, but like just being very honest about what it's trying to do, what it's trying to envision, and that it really genuinely wants there to be a better tomorrow, and that that this group of people is trying to not manipulate but show, and let people decide for themselves, that kind of thing. So I think that something like that could be a valuable contribution. I would love to see the science fiction writing that translated to film, some way of finding ways to get it to the masses, get the type of science fiction to the masses that is really a lot more about thinking, a lot more about seeing the wonder, a lot more about contemplating possibilities, and less about.
Seems like we lost Logan for a moment, but I would definitely encourage our viewers to read his post on rational romanticism, and hopefully we will get back Logan's connection soon. Looks like he's attempting to come back. And in the meantime, I'd like to highlight this comment from Carlos Neto, who writes, "Science fiction is very powerful. All history as we know is fiction. Somehow it is impossible to be 100% sure." I think he writes this in reference to historical events and there being some element of fiction or perhaps speculation or educated hypotheses in narrating historical events. Alan Crowley writes, "Does fiction influence society, or does society influence fiction?" And the answer to both of those is yes. And John H. writes that space travel and time travel were pretty much the whole enchilada back in the day. And he refers to famous science fiction authors of the Golden Age: Arthur C. Clarke, Isaac Asimov, Robert Heinlein, Ray Bradbury. Ben, in our internal chat, points out a fairly common statement that history is written by the winners. And we are still trying to get Logan back, but we are not yet able to see him. Nonetheless, his comments have sparked a lot of fascinating reactions within the chat. And Carlos Neto writes that he thinks it is a great idea to add rational romanticism on the transhumanism table. And he thinks that there's a possibility that this would help to add more women to the transhumanist movement, as they are more than 50% of the human population. Logan, we have you back. Thank you for rejoining us. And please conclude the thoughts that you had in regard to science fiction, and then we'll see if we can get in another question from Art Ramon.
Great. Sorry about the internet interruption there for a moment. As I was saying, I think that the translating science fiction into film that is more thoughtful science fiction and less defeatist science fiction is definitely a challenge of the age. And seeing how we can find ways of convincing society to value the type of optimism that was maybe seen in the 80s in particular, as well as the 50s. Obviously, there was the New Age stuff in the 80s, but I think in film there was more optimism at the very least. But in the golden age of science fiction, maybe the film of the 80s, and so on and so forth, I think that there was definitely a lot of a lot better of an attitude and something that was more constructive. Some examples from more recent, I guess Ted Chiang comes to mind in terms of science fiction that is really, really thoughtful, really in touch with humanity, and is not entirely dystopian, but really encourages us to have a sense of wonder about the future and the possibilities while still thinking about all of the ins and outs in a very fascinating and in-depth way. Ted Chiang's story, the story of your life, was made into a movie called Arrival that was in the theaters, and I would love to see more things like that come out. So yeah, with that, I'll wrap up my answer to this question. Like I said before, I would like to see a more organized effort towards a transparent yet organized effort towards getting science fiction to sort of reclaim some of its sense of wonder and cast away some of the defeatism.
Excellent. And it is a hope that I very much share, as do many members of the U.S. Transhumanist Party and the transhumanist community. So we have about 4 minutes left in our virtual Enlightenment Salon. I would like to offer Art Ramon Garcia the opportunity to ask a question, and then we will go into concluding comments. So Art Ramon, please go ahead.
Yeah, speaking of dystopian, I heard some report from somebody that the Russians had some biowarfare, either genetically engineered bird flu or coronavirus, where they specifically targeted Anglo-Saxons. Do you think that's an actual possibility to target one specific group, or is it just disinformation?
I don't know if that particular report, I can't really comment on that particular report, but I do think that we're going to need to have better biosecurity measures in place in the future to prevent people from engineering bioweapons that could be broadly destructive or even destructive to specific groups. There are I do think it's a possibility. I don't know if it necessarily would be by race. That might be a trickier problem or a trickier obstacle to overcome in terms of engineering, but there's definitely a possibility that people could be targeted by certain genetic markers, and I would be concerned that that would be something that might be used as a proxy for certain problematic practices. So that said, I think that technology is always sort of competing with itself. And so we have this world where people are very much in sort of this race against each other to keep the evolutionary cheaters from doing something that is problematic. And so I do think that the in parallel advances of biosecurity and other technologies may be able to circumvent any kind of super widespread destruction from bioweapons. That said, it's still a concern, and I think we should be making every effort to combat that threat.
Yes.
Thank you.
Thank you for your response, Logan. Personally, I think it would be quite infeasible to develop any sort of bioweapon targeting a particular ethnic group, because ethnic groups are not monolithic. Throughout history, there has been so much mixing of populations. There's a ridiculous conspiracy theory that occurred during the early stages of the war between Russia and Ukraine, where some Russian government propagandists were accusing the Ukrainian side of developing bioweapons to target people who were ethnically Russian. Well, the problem with that, even if they had done something like that, would be that it would kill or hurt most of the Ukrainians as well, because of how much mixing of those two populations there has been. So I'm a bit skeptical as to that possibility. Our friend Alan Crowley wonders, Logan, if you are willing to collaborate on science fiction shorts, novellas, or novel-length works, and also any suggestions on starting a pro-science fiction group. So perhaps in the last minute of our salon, if you could share any thoughts about that, as well as any other concluding remarks that you would have for today.
Yeah, so I'd definitely be interested in trying to get some type of pro science fiction group going, some type of organization, even if it's just the seeds of it for now. It might need some time to sort of grow and mature, but some type of science fiction organization that is attempting to do the things that I described, maybe even if we're just planting the very beginnings of it, or the very beginnings of one of such organizations, I would be interested in speaking more about that. As to collaborations, I do want to let you guys know that I have a lot of projects going on. As I mentioned, I have 3 different projects for my PhD. I have the connectomics work, and I have some work at Conduit still ongoing. So I am stretched a bit thin at the moment. However, I'm always happy to discuss specific ideas, and I'm always happy to have Zoom meetings with people if they'd like to talk about more, or even just texting or calling or whatever, and fleshing out whatever in particular they might be interested in, and seeing if it can fit within my bandwidth. So I always have more things I want to do than I actually can, but it's a forever struggle. But I do enjoy. I definitely do enjoy helping out with things like that. So if anyone wants to talk about specifics, I can certainly consider it.
All right. Thank you very much, Logan. And we really appreciate you spending nearly 2 hours with us today on a virtual enlightenment salon that was as wide-ranging and interdisciplinary as I had hoped it would be. And we wish you all the best in your future endeavors. And to our panelists and our audience, I hope that you all live long and prosper.